From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 4, recorded on December 22nd, 2015. Hi everybody, I'm Vincent Racaniello, and you are listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels Elde.
Good afternoon, Vincent. Good to hear you.
Our episode 4, we're just moving on, aren't we?
Yeah, we're doing it.
And we have a guest, a special guest, also from Salt Lake City, Nitin Fudness. Welcome, Nitin.
Hi, Vincent. It's lovely to be here.
So I have Nitin. Is that the right way to say it, or should I do something differently?
No, you got it right. Nitin Fudness.
Nitin Fudness. And his most unusual name, and especially since I understand you originated in India, correct?
That is absolutely right.
What part of India?
I'm a West Coast guy, so I was born in Bombay.
West Coast.
And I grew up in a city called Pune, actually, which
sure.
I still miss it.
Pune is the technology hub, right?
It is, an education hub.
Yeah, my wife worked for Merck for a long time, and they had some collaborators there, so she went to Pune a couple times.
I hope she enjoyed it.
She loved it. She loved the food.
That's one of the best.
Particularly, yeah. But she's no longer going there, so... But I remember her going very well, because she left me with my kids, you know.
I see. Well, hopefully next time I can take you to Pune. And
I've never been to India, man. I'd love to go.
I'd highly recommend it.
You've been, Nels?
I have. So I spent some time in Bangalore, and then at a meeting on evolutionary cell biology a couple of years ago.
Mm-hmm.
And then more recently at a meeting in Agra, just in the shadow of the Taj Mahal. It was really incredible.
Cool. That's neat. So Nitin, how old were you when you came to the U.S.?
I came here for my PhD studies, so early 20s.
Early 20s. And where did you do your PhD?
I did my PhD with Alan Orr at University of Rochester in upstate New York.
Right. I visited there a couple of years ago. They gave me a named lectureship or something.
Nice.
And I had a good time. It was the students who invited me, basically. But it was February, really cold.
Yeah.
You remember Rochester winters?
I do. It's mostly a blur because every time I would go into the lab, it would be dark. By the time I came out, it would be dark in the winter. And then, of course, you spend some time shoveling your car out of the snow.
Yeah. Now, if I'm not mistaken, you did a postdoc next with Harmeet Malik, right?
That is correct.
Am I saying it correctly? Harmeet, or is it Harmit?
It's Harmeet Malik.
Malik. And he went to Rochester too, right?
That is correct. We were, in fact, and got our PhDs from the same department. And we didn't overlap.
Yeah.
But I did a lab rotation in his PhD advisor, Tom Eichbusch's lab.
Right. Wow, that's something. Did you go there because of him, or you learned of him once you were there?
I learned of him once I got there, because Harmeet becomes famous wherever he goes.
So is that why you ended up doing a postdoc with him, because you learned of his work while you were at Rochester?
That's one of the things. I mean, he's just a person with such great goodwill and such amazing record of mentorship.
Yeah.
But also,
with the exception of Nels, of course.
That's right. Present company excluded here.
Yep, Nels was still in the lab, in Harmeet's lab, and actually that was also one of the wonderful things that made it super clear to me that that's where I wanted to go. I mean, there just aren't that many people out there who can bridge the fields of evolutionary biology and cell biology the way that Harmeet does. And so really, if that's the zone you're hoping to be in, your options are fairly limited today.
Wow. So what did you work on as a PhD student?
So as a PhD student, my major work was on understanding speciation in a pair of species of Drosophila pseudoobscura, and to understand how hybrid sterility evolves between these two subspecies, And in the end, the answer turns out to be that there's a lot of selfish gene dynamics that plays into how these sorts of phenomena evolve.
That's a similar story as what you did as a postdoc, right?
Similar and different, and I'm really excited to talk about it.
Yeah, so we're going to talk. So this paper we're going to talk about was done during your tenure with Harmeet, and we'll talk about that. We have a few things to catch up on first, right, Nels?
Yeah, that sounds good.
First is the weather, which I understand is really interesting in Salt Lake right now.
Yeah, we're in the middle of a blizzard, which as a native Minnesotan, this is exciting times. It's really fun. I think I was just looking at the ski resorts, and they're getting on the order of 2 inches an hour of snow right now.
Wow.
And they're counting the totals in the feet and not the inches. And so I think there'll be some skiing in the near future.
Nice.
Yeah.
Well, right now, it's dark out. It's almost sunset here. And it is 14 Celsius, and it's raining.
I heard it's kind of tropical conditions for the holidays in New York.
Yeah, it's been pretty warm. Actually, yesterday and the day before, it went down to freezing, but now it's warmed up again. It can't make up its mind. And it is it's black and cloudy and pretty nasty out. It's a good day to stay inside and do a podcast.
Yeah, cozy up by the microphone.
That's absolutely right. Yeah, I'm getting my podcasting done early this week because it's holiday. Nobody wanted to podcast on Friday, which is Christmas Day.
Yeah.
I would have been happy to.
Well, you're kind of a natural podcasting athlete, I would say.
Hey, there's got to be a word for that. A castleet.
Yeah, there we go. Maybe work that one out a little more.
I just love talking science, and I can do it from home as well, and I would have done that, but my co-hosts on TWiEVO said, no, we can't do it on
Friday.
So we have to do it tomorrow, which is a short turnaround because we just did it on Friday.
Oh, boy. Yeah.
But, that's okay. And I'm trying to figure out when we could do a TWiM also. It's a little tough around these 2 weeks,
No kidding.
But we'll do that.
Folks are traveling. Yeah.
I've finished traveling for the year.
Mm-hmm.
I have to tell you, a couple of weeks ago, I visited Mississippi State.
Oh, wow. That must have been fun.
It's in Starkville, Mississippi. Starkville. What a name for a town.
Yeah, no kidding. Is that near Jackson?
Yeah, it's not too close to Jackson, no.
Okay.
No.
It's the only town I'm familiar with from my travels.
But I visited a biology department, which is always interesting because, I guess, are you in a bio department also there, Nels?
I'm in human genetics,
genetics.
But our guest, Nitin, is in the biology department here at Utah.
Yeah, that's really one of the few places where you can work on fruit flies and other interesting things, So this department in Mississippi State was biology. So I had talked with people working on amoeba, prickly pear cactus.
Wow.
Bacteria, of course, and a variety of other things. So you can do, and one guy did population studies on various seed crops, that sort of thing.
Yeah.
All the stuff that NIH does not fund, you can work on the biology department.
That's right. Although, as I think we'll be hearing some from our guest today, I think, and with this, this synthesis that we've been talking about on TWiEVO between evolution and experimental biology, some of the diversity in the ecology from the field is now coming closer and closer to the laboratory and to experimental approaches that actually do get NIH's attention.
Absolutely. And during my visit, guys, I gave a talk there. It was one of these named lectureships, right? Where
very cool.
After someone, and the name of the lecture was the Lewis Brown Lectureship Award, and it was presented by the local ASM student chapter. And the plaque they gave me is not really a plaque; it's a cowbell.
Oh, is that right? I think I've heard it in the distance.
Yeah, here it is. You hear that?
Yeah, I love it.
So, the cowbell is, Mississippi, they have a lot of cows.
Of course.
And during the football games, apparently everybody brings a cowbell and they ring it.
Wow.
And they had to actually stop them from ringing during the plays because they were disturbing the visiting team.
I see.
So they can only ring it between plays. And it's very loud, apparently, because they have 60,000 people in the stadium.
Oh, man.
Right?
Oh, yeah. That's going to add up fast.
This is one of the nicest plaques I've ever received.
Yeah, what a neat souvenir.
So a couple of, on TWiEVO I mentioned and I rang this thing. And so we got immediately a letter that week from someone who said, there are some people who feel, actually it was a tweet, some people feel TWiEVO needs more cowbell. Do you know that reference?
Yeah, well, now you're connecting Mississippi to New York City and Saturday Night Live.
Very good, Nels. I'm impressed. Well, it's a good thing I decided to podcast with you. You got it all down. Yeah,
well,
the episode of Saturday Night Live. So we were talking about it this past week, and we started talking about songs that have cowbells in them, and immediately came to mind this song by a group called Mountain, which you probably have never heard of because you like bluegrass,
Correct, yeah.
And it was called Mississippi Queen, and it starts out with a beautiful cowbell. And
wow.
I appended a clip of that at the end of last week's TWiEVO.
Ah, nice. I haven't caught up with that one yet.
Yeah, it's okay.
I'll take a listen.
What have you been up to, Nels?
Well, I've been out on the trail myself, too. So I was a couple weeks ago up in Seattle, Nitin and my old stomping ground. But I was at the University of Washington, not the Fred Hutch.
Mm-hmm.
The graduate students in the Department of Microbiology invited me to give a lecture. And I got to tell you, it was just really fun. Great energy there. They've got some really good traditions. So you give the seminar, and then immediately after, go to the old College Inn, which is a small hotel just north of campus, and has a great old fireplace.
No, I've been there.
Yeah. So that's the old tradition. And we were there, drinking beers and eating nachos and kind of taking the conversation from the seminar in different directions for
nice.
a long time. And so, yeah, it was really a blast. And then, so that was a fun one. And I turned around pretty quickly, and I was just in San Diego last week at the American Society for Cell Biology meeting.
Nice.
I got a small plaque myself. It's not a cowbell, but a pointy piece of glass as one of the first finalists for the Gibco Emerging Leaders Award in Cell Biology.
Cool.
And this was really fun because it was a chance to kind of make the case to some of my colleagues in cell biology and also some of the leadership in that society about the importance of evolution and bringing in those ideas to the cell biological approaches. And I think more and more people are starting to recognize this, that by considering the diversity of life around us and taking more comparative approaches and thinking about some of the deeper questions that we'll be getting in today with Nitin, that there are some really exciting new opportunities to kind of reshape some of the foundations of some of the major approaches to biology.
Part of what we do here on TWiEVO, right?
That's what we're going for. Yeah, exactly.
One more thing I have to update you. About 2 weeks ago, my last PhD student defended his thesis.
Hey, congratulations. That's very cool.
So I've had 24
wow.
PhD students graduate in my years here at Columbia. I have been here, 33 years.
Wow, that's impressive.
All in one place, yeah.
Yeah, what a great legacy. 24 students. My goodness.
And that's it. I'm done.
Well, I wouldn't say quite done. It sounds like your podcast schedule, if anything, is only picking up steam.
Yeah, I'm done with students. I don't want to train anymore because I want to phase down the laboratory slowly, and then focus on podcasting.
Yeah.
Which I think is not only fun, but makes an impact.
Yeah.
Now, you and Nitin are buddies, right?
We are. So as Nitin kind of alluded to, we first crossed paths as postdocs in Harmeet's lab. And correct me if I'm wrong, Nitin, but I think we overlapped for about 2 years.
I think that sounds about right. A little bit more, maybe.
Mm-hmm.
So you have here in the show notes, Nels and Nitan. It sounds like to me like a law firm, guys.
That's correct.
A science law firm.
If it doesn't work in science, you can put up a little... what do you call the sign that they put up?
A little shingle.
A shingle.
There we go.
Nels and Nitan. That's great.
Well, I have to say, Vincent, so we have taken our show on the road once or twice. When we were still postdocs in Harmeet's lab, we put in for a symposium at the annual Society for Molecular Biology and Evolution meeting, and we were chosen to to lead a symposium on molecular arms races. And I think I'll let Nitin speak on this for us in a second, but one of our main goals, in addition to the fun biology and picking some of the speakers and highlighting some of this work, was to introduce each other at the podium. We thought it would be kind of fun to call each other our esteemed colleagues and try to give flowery introductions.
Yeah. So, I mean, that's really funny because, in a sense, when we saw the notice for the first time, that was the entire motivation, that it would be hilarious to just be able to introduce each other as our esteemed colleagues. And so it's kind of cool to have a throwback today where I kind of missed Nels not calling me an esteemed colleague. but it's good to be here, esteemed colleagues.
Well, you're a special guest, you know.
That's right.
Some people like that, too. So you guys were really friends, right? You weren't just postdocs together. You used to do stuff outside of the lab?
Exactly.
Yeah,
good. And that's really, for me, I mean, this is what makes it all worth it, is in addition to the science, is all of the personal connections and just, friends for life. I mean, it's really rewarding on multiple levels.
So you guys are now amazingly in the same department.
Actually,
the same institution, right?
Same institute, that's correct. Different department, same institute, and it's kind of cool because we really are extremely close friends. But we bring really different backgrounds and different perspectives, even different training, but sort of unified by our love for science. And
was Nels a factor in your going to Utah?
He certainly was. I mean, Hermite's lab was at the top of my list. In fact, the only place on my radar to go to. And once I interviewed there, I was just stunned and blown away. And that was the beginning of a real lifelong and deep friendship.
Mm-hmm. How long have you been at Utah now?
I've been here for about 2.5 years now.
And Nels, you've been there 5-6 years?
I've been 4.5 years, but who's counting, right?
Yeah, right. But Nels must have, nudged you to go there as well, right?
Oh, absolutely. I mean, he was a bigger factor for me to come here, in fact.
Yeah.
And I knew that if Nels was here, then this place had to be amazing.
Well, and I have to say, so if we just look at the landscape here at Utah, in addition to the medical school in the genetics department where I am, and also biology, there's great evolutionary stuff happening here. So Michael Shapiro's lab, who's actually doing pigeon genetics, which may come up late in the hour here. And then folks like John Seeger and others. And so there's this really great tradition of both evolution and Drosophila work. I'm thinking of labs like Kent Gulleck's and others. And so it was kind of a perfect landing place for Nitin as he was starting out his lab.
So we could also tap those individuals for TWiEVO, right?
Yeah, I think we certainly will. We'll also, I don't want to make it sound like all of the fun research is happening in Utah, although maybe most of it is. But no, we'll be kind of going all over the map to chase down exciting science at the interface of experiments and evolution.
Well, here in New York, we have a fondness for pigeons. So I'd love to hear a pigeon biologist talking about them.
Yeah,
I want to know why they're missing feet or toes,
Exactly. No, it turns out to be really fascinating. And this is exactly what Mike's lab is doing, taking a genomics approach to the artificial selection that's been imposed on pigeons over hundreds of years, and some really cool findings. So that'll come up down the road.
Couldn't you also explain why their heads are wired to their feet?
He can explain many things, some of which are not even sort of suitable for polite company when it comes to pigeons. All kinds of crazy features.
Now, you two can actually see each other from your respective offices. Is that correct?
Yeah, so let me give you a little bit of the landscape of Elde Lab Studios here. Just from my office, which is Elde Lab Studio Room 1, is a conference room, Studio Room 2, where Nitin is podcasting this afternoon.
So he's in a conference room next to you. Okay.
Exactly.
But his department is farther away?
It is. So the Department of Biology is on main campus. So turns out the University of Utah started, I think, back in the late 1800s. Do you know the exact year, Nitin?
I don't know the exact date. Something like that. It was a while ago.
You're right next to the plaque that says it, so I thought maybe it was seared into your memory. But so that's the main campus. And then what's happened is over the years, as the medical school came online, they kept building higher into the foothills of the Wasatch Front. And so we're at about 500 feet higher elevation here in the medical school genetics department.
So Nitin had to walk to get to your conference room?
I had to walk to my car.
Oh,
it's snowing outside.
You drove. It was probably a 5-minute drive, right?
Yeah, but less than that.
Oh, okay. Well, wonderful. This is great. It's good to have good scientists, but when they're buddies, that's even better.
Yeah, this is fun, and I'm really excited about both today's topic, which we just hinted at briefly, the idea of speciation genetics, or how one species can become two. This is sort of the genetics of reproductive isolation that Nitin is one of the world's experts in. And actually a question that goes all the way back to Charles Darwin. And when he was considering this idea of species diversifying and then not being able to reproduce, it was really baffling to him. He even dubbed this the mystery of mysteries.
Mm.
And so that's what we're going to dig in on today is speciation genetics, which was not only being thought about 150 plus years ago, but continues to be an active area of research. And Nitin, along with Harmeet Mullick, just published a paper in the current issue of Science, which sort of answers one of the long-standing major questions about speciation in fruit flies and Drosophila.
You know, I saw this paper in my literature searching, and I said, boy, this is cool. I wish I had a podcast to do it on. And I said, I wonder if Nels would be interested in this.
Yeah.
I didn't know Nitin at the time. And then you turned out to, well, do it. So that's pretty cool. It's called An Essential Cell Cycle Regulation Gene Causes Hybrid Inviability in Drosophila. And I wonder, Nitin, if you could start by telling us what is a species?
So that is actually an interesting question. And the currently accepted and most useful definition is something called the biological species concept.
Mm-hmm.
And according to this idea, species are interbreeding populations that don't breed with other such populations. So the idea is that these are populations which have distinct gene pools and don't really exchange too many genes across populations,
Mm-hmm.
essentially giving them independent evolutionary trajectories through time.
Okay, so Homo sapiens, that's us, right?
Yep.
There's nobody around for us to breed with. Well, some people try to breed with animals, but they don't give offspring. But didn't we breed with Neanderthals at one point? What were they?
That is actually a really exciting finding from these last few years, which is evidence of gene flow across populations between Neanderthals. And so again, one of the interesting points is that species can also be a continuum where early during speciation events, you could probably still have some gene flow,
I see.
which eventually is a tap that gets turned off afterwards.
What's the genus species for Neanderthals? Do you know?
I don't actually.
Let's look it up.
Yeah. Is it Homo Neanderthalis? That's my shot in the dark guess.
It is Homo. I'm on the page here. Homo Neanderthalensis, right?
Ah, close.
Yes, that sounds correct.
So it's a different species, same genus, right?
Mm-hmm.
Yep.
So that, so maybe it was early on that they were able to do that. The reason I ask, Nitin, is because in viruses, people call viruses species, but, viruses don't really have sex. They recombine in an infected cell. But the species concept really doesn't work for a virus.
That is exactly right. So that is actually one of the caveats of the biological species concept, which is it doesn't quite work for things like bacteria or viruses. But again, I think from an evolutionary biologist viewpoint, recombination and gene flow is sex. And so I think some viruses might be upset at you saying that they don't have sex.
Well, what's sex? Let's define it. That's simply the exchange of genetic information between two species.
Yep, that's exactly right.
Yeah, I think that's a good definition. And just to point out, some of the systematics, the genus and species definitions are occasionally clouded by some of the points that Nitin is raising about gene flow and sort of the last vestiges of that. And can you remind us, Nitin, about some of the details of the different types of speciation events, basically the geography versus the compatibility?
That's correct. So one of the sort of classic points in speciation biology is the idea that you first need what is called allopatry, which means for populations to be geographically isolated. And now that initially they're geographically isolated, they can now evolve in a way that eventually, if they were to meet the other population again, then something would be wrong with the hybrids, or there would be some other barrier to gene flow, either behavioral or even after a zygote is formed, it could be sterile or dead. And so these barriers are essentially the key to understanding speciation.
So the incompatibilities between species can be manifested in different ways. You just said that. It could be you never get offspring, or you get offspring that are sterile, right?
That is correct. If you go back in history, even a long time ago, Aristotle noticed that if you cross donkeys and horses, you get mules, and they're viable but sterile, or mostly sterile. Mm-hmm. And so that is, again, an example of this sort of incompatibility responsible for hybrid sterility.
So that's a case where the offspring is useful nevertheless, right?
That's correct.
Are there other examples of, aside from the system you work on, are there other kind of well-known examples of incompatibility?
Oh, absolutely. I mean, there are thousands of examples. One favorite example that I sometimes like to talk about in my seminars is that of lions and tigers.
Okay.
And you can get ligers that are sterile. And in fact, one of the cool things is that the biggest cat is not the tiger. It's in fact the liger.
Interesting. So the whole concept of speciation, that is the arising of new species. This is how evolution works, right? Because, as you said, a species will move to two different geographical locations. They will evolve separately, and then at some point you have a species arising. So speciation is response to selective pressures, basically, right?
So that part becomes a little bit trickier. Mm-hmm. So what role does selection play in terms of causing the birth of new species? And so for example, you can think about things that are called as ecological speciation, mm-hmm. where, for example, if the two populations adapt, in fact, with selection to separate niches and separate environments and become specialized, and now you form a hybrid that is unfit in either of those environments. That is one of the clear classes of speciation where selection is directly responsible. Mm-hmm. But when it comes to intrinsic barriers, such as hybrid sterility or hybrid inviability, where there's something intrinsically wrong with the cell, it used to be very hard to say whether selection was responsible, but now we're starting to get some answers.
Okay. So are we, are new species arising today?
Oh, all the time.
Like where and when and how?
So again, this is actually one of the conundrums of being an evolutionary geneticist, which is our science in the wild is happening at timescales that are not suitable for our lifespans. Mm. And so we do have examples of sort of new species arising when you look at real-time evolution. And in fact, some of my PhD work was focused on these species called Drosophila pseudo-obscura. The name itself sounds like, why would anyone work on a species that has the word obscura in it?
Exactly.
And the key to that is trying to go to solve a problem where the problem should be solvable. And in fact, in these paraspecies, they are very early in their speciation event, where only one direction of the crosses yield sterile hybrid males. And it is, in fact, really hard to find these cases where speciation can be caught in action, and then you can go back in, do some genetics, genomics, cell biology in the correct context. And in fact, that's why you go to species that have the word obscure in their name.
Mm. I guess so the best example I know of is in microbes, bacteria, and viruses. You just look and you find ones that you didn't know about before.
That's correct.
But if you move up to larger organisms, as you say, you need to wait a long time for a new species to emerge, right?
Correct.
Because is it fair to say that? Actually, I should pose it as a question. Do we know of all the animals on Earth right now? For example, we'll put it at animals. We won't make it harder.
Right. That itself is debatable, right? For example, you can even now find a new species of whale in the ocean, and that sort of discovery is stunning. And so I think we do keep finding more things all the time. And even a good estimate of how many species are there on Earth itself is a huge open question.
And so if you find a new species, it's probably because you hadn't noticed it before. It's not that it had just emerged in the last few months or years, right?
That's correct. That's correct.
Nels, you were going to say something.
Yeah, so just to echo a bit on this point that, I think when people discover a new species of animal, it is a really exciting thing. And it's, for me, or my impression of it is that it's just sort of sampling, that when we look at microbial populations, we just have sampled so few.
Yeah,
one of my favorite examples are these giant viruses that people are pulling out of marine environments or water cooler towers. And there's one in particular a few years ago, and I know you've covered these guys a lot on TWiEVO, Vincent, where they, I think, caught this virus off of the coast of South America, did a blast search, and saw that something like 95% of the open reading frames were never seen before.
That's right.
Yeah, it's just so exciting to think about all of the diversity out there that we have barely scratched the surface.
Yeah, I just had a thought which has left me, unfortunately. Let's see, we're talking about species. Oh, well, it's gone. All right, so this paper of yours deals with, again, Drosophila, but two different Drosophila species. So maybe, Nitin, you can give us the background on this story.
Sure. This is actually one of the classic and celebrated cases of speciation genetics because it involves Drosophila melanogaster, one of the first and most sophisticated genetic systems that we have as geneticists. And so the story is actually quite long running, and I can give you a taste of how old this problem is, which is actually exactly what makes this really special for me, which is to work on a 100-year-old problem that all of my heroes, including Ed J. Muller and so on, have worked on it, is really special. So the story starts in 1910, which is actually right after the rediscovery of the principles of genetics, where there was a master student in Thomas Hunt Morgan's laboratory, who showed that, man, if you do these crosses, you only get only get females out, and you never get any adult males. And there's a really nice paper called Unisexual Broods of Drosophila. And you can imagine how confusing it must have been to be there right at the rediscovery of Mendel's laws to see this kind of thing show up. And so it took another decade or so for another giant in genetics called Sturtevant to show that this is actually an interspecies cross. In fact, what Quackenbush, who was a master student, and he actually unfortunately later died of a disease, I think it was tuberculosis. But Sturtevant picked it up and showed that this is actually a different species called Drosophila simulins.
So they didn't know that there were two species that they were.
That is exactly right. So you were just mentioning discovery of new species, and this is again another example of how a new species was discovered. It didn't just speciate, but we just observed this.
So Nitin, let me just interrupt you a second. I'm going to do this all the time. I'm sorry. So today you would just sequence the genome, right? If you found some new organism.
Correct.
Could you tell from the genome if you had a new species?
That is hard to say. So what you're essentially getting at is if I were to compare a new genome sequence with a known genome sequence of a known species, if I were to have some sort of metrics of divergence, can I say that these are different species? Actually, the answer is not that simple because different taxa speciate at different levels of divergence. So there are some critters that will actually still breed with each other despite a lot of divergence. And then there are others where you get just a little bit of divergence and that's enough to produce new species. So really, that is, again, one of the unfortunate things, and also some of the coolest. What makes the biology really cool is that just sequence gazing is not really going to get you too far.
Okay, so these guys, Sturtevant, you said, found that they were two different species. Is that correct?
That is correct. So he pointed out that what Quackenbush was doing was actually an interspecies cross where the hybrid males are dead. And he published this really incredible series of papers outlining the problem. And since then, it's kind of been a collective obsession to sort of figure out why are these hybrid males dead? And that's when a lot of people in Thomas and Morgan's lab even took cracks at it. Dobzhansky, who's another father of evolutionary genetics, actually got quite close, and he used triploid melanogaster females and crossed them to simulins males and so on. But the real breakthrough came when Ed J. Muller, Herman Muller, again a Nobel Prize winner, used X-rays for all his experiments. And with another famous geneticist, Guido Pontecorvo, they did these incredible experiments where they took triploid melanogaster females, crossed them to heavily irradiated simulins males to make these sort of pseudo backcross progeny to figure out what genes or what chromosomal regions are involved in this hybrid inviability. What can one say about the architecture of that hybrid incompatibility that kills these hybrid males?
So it's interesting that they by that approach, I guess they were assuming it would be one or a few genes at most, right?
You've again hit on a wonderful point, which is it wasn't really clear if this was one of those things that has 1,000 different loci or small effect that act additively.
Right.
And in fact, these are the exact kind of experiments that Dobzhansky and Muller and so on, that was the objective. And through this, they could show that actually it is not 1,000 genes of small effect. It's actually a complex interaction between a few genes.
Yeah, they were lucky, right? If it were 1,000, forget it.
That's correct. That's correct. Good luck with the genetics. Huh. That's a long thesis project.
There you go. And just to point out really quickly that a lot of this historical work that Nitin's describing was happening right in your backyard there, Vincent, at Columbia University.
That is correct.
That's right, yeah.
In the fly room.
The fly room, which they still have here. You know, you can visit it. It's like a museum.
I would love to. I would love to.
Yeah, I mean, someone told me recently, and you can make an arrangement to go. It's there. It's closed up. You know, it's not open to daily traffic, but you can make an appointment to go.
Wow,
cool.
Yeah, it'd be cool, right?
Yeah.
Maybe we should do a podcast from the room sometime.
Oh, wow. Now we're talking. I like that. Great idea.
So what did they identify, Nitin?
So essentially, I mean, in a nutshell, what you could say after all those experiments was there is something on the melanogaster X chromosome that interacts simultaneously with something on the simulin second and third chromosomes to kill the hybrid. Now, if you were to substitute even one of those incompatible partners with a compatible one, for example, if you took that melanogaster X chromosome that is required and substitute it with a simulins X chromosome, that hybrid is now alive. And so you really need all three components to be dead. And so that was actually one of the critical conclusion from those series of papers, including from Guido Pontecorvo's work.
Did at some point the specific genes get identified? And if so, how?
That's correct. So, I mean, I guess Muller and Dobzhansky and all those guys had no hope of ever finding the gene because it wasn't even clear what a gene was in those days. But they did answer that it's just a few factors that are responsible. And so, actually, there was a long stasis in this field until one amazing breakthrough where strains from Drosophila simulins could be isolated that when crossed to melanogaster produced viable hybrid males. So people called this a rescue strain. So for example, the simulins one is called lethal hybrid rescue. And then similarly, people just went back and people like Ashburner and Watanabe were huge players in this, where essentially you go and you collect hundreds of melanogaster strains, and then you do the same cross, and then see if is there a particular strain that makes viable hybrids instead of dead hybrids. And in fact, two such strains or multiple strains were identified, some in melanogaster, some in simulins. The melanogaster one is called hybrid male rescue. The simulins one is called lethal hybrid rescue. And once you have these strains, you can then breed them and do crosses within species. So melanogaster HMR cross to melanogaster strains that make lethal hybrids, and use this sort of approach to map and identify these genes. So both of these strains and the genes responsible for their rescue effect were identified by Dan Barbash's group at Cornell University through some beautiful work, and really showed that these are the genes. And in fact, the gene names are also called HMR and LHR, which stands for the same strain names. So two of these three guys are now identified. And the question was, what about the third gene?
And this identification of the two are relatively recent developments, right?
That is correct. So in the early 2000s.
So we have two genes and a third. And is that when you come onto the scene?
That is exactly right. So one of the questions here is why did this take so long?
Yeah.
And in fact, like I said, there was a huge breakthrough in the early 70s to identify or to even see the existence of these strains. But after screening for all these rescue strains, they all either belong to HMR and melanogaster or LHR and simulins. And there really wasn't another strain that could correspond to a third different gene.
Mm-hmm.
To make matters worse, you cannot do regular recombination-based mapping between melanogaster and simulins because all hybrids are dead or sterile.
Mm-hmm.
And so if you think of it from a geneticist viewpoint, genetics is the art of inferring things by doing crosses. And speciation, by definition, means you cannot do crosses. So this becomes a methodological paradox where speciation genetics becomes an oxymoron because speciation, by definition, means you cannot do crosses.
So you would normally take a pair and cross them and then take the offspring and do further crosses, either back crosses or with each other or whatever, right?
That's exactly right. But you can't do it here because everybody's dead or sterile.
Yeah.
Yet another problem is whatever kills these hybrids is a dominantly acting factor from Drosophila simulins. And so we have the whole toolkit in Drosophila melanogaster, but it really doesn't help us identify this gene because what you would really want is a sort of deficiency or a deletion toolkit in Drosophila simulins, which does not exist. And it's really hard to make it too, or really difficult. What one would do is to try to reinvent the whole melanogaster toolkit in Drosophila simulins, but we don't even have really basic tools, for example, things that are called balancer chromosomes that you could use to maintain mutations.
Mm-hmm.
So for all these reasons, essentially, all traditional genetics approaches basically hit a roadblock to identify this gene.
So did people stop working on this problem?
It's hard to say stop working. I mean, it's one of those things that lies in plain sight.
Yeah.
And people are working on it. But this identifying the third gene, in my opinion, was one of those things that even I knew about as a young graduate student.
Mm-hmm.
And again, in this paper, the methods that we use are again, individually, each step is really simple. And that's what I love about it. But it just takes some time to connect to these open, under-your-nose problems that are obviously important, long-standing, and then to connect it with a way to sort of weasel your way out of these traditional barriers.
So this was a problem you tackled as a postdoc. I want to know how that developed. Did you go there and say, hey, Harmeet, I want to work on this long-standing problem, or what happened?
Not quite. I mean, it's a little funnier than that because I mean, in the Mullick lab, it was kind of fun for me to come in and look at this broad open space of genomics and cell biology and evolution, and I kind of was playing around with actually a handful of ideas. And as is my wont, I would often go around saying, hey, you could solve it this way, and just keep doing it.
So that's right. And I can remember a lunch or two sitting across the table from Nitin in the Fred Hutch cafeteria and him saying, hey, you know what, this problem's been around for a long time. And then after, I think, sort of being in the Mullick lab environment for a while, and as Nitin mentioned, seeing kind of all the tools and possibilities, the sort of solution or the idea for how he might actually get some traction on this long-standing problem came into focus. And the trick was the genomic possibilities that were just coming online with the technology.
That's correct. In fact, Nels was there, and he can attest blow by blow how this played out. But essentially, I had to go to Harmeet and say, hey, one could solve this problem if one did this. And Harmeet's response was, oh, why is the one not you? Just do it. And that's actually how it began, by just sort of a casual banter and egging each other on.
So what was your idea for cracking this?
So the idea is actually so simple, and like I mentioned, that's what I love it, which is the idea is that we know that there are certain genes in simulins if you knock out, for example, LHR if you have a null mutation, and if such a sperm fertilizes a melanogaster egg, that hybrid is viable. So the idea was very simple. What if we ran a mutagenesis screen, and if by chance you have a sperm from Drosophila simulins that has a mutation in another gene that also makes the hybrid viable, you would get a hybrid male when normally there just isn't any. Mm-hmm. And so
it is very simple.
that is correct. But again, each single roadblock becomes very simple. And as Nels was alluding to, this was one of the times where sequencing two entire genomes from a single hybrid fly just became possible. Mm-hmm. So if you make a hybrid between melanogaster and simulins, and if you sequence both genomes from one hybrid fly, we can now go back and ask, who is the melanogaster part of these sequences? Mm-hmm. Who is the simulins part? And who are the new mutations in the simulins components?
Right.
And so if such a sperm with a mutation does fertilize a melanogaster egg, you get that viable hybrid male. And as I mentioned, that male is sterile. So you just get one tiny fly. Mm-hmm. And there's really no making a stock or using it for anything. The only thing you can do with that sterile male is crush it and sequence its DNA. And if you did that, you will still see hundreds, if not thousands, of new mutations in the simulins genome of that hybrid male. And so you will come down from, 10, 13,000 genes to maybe 1,000, so close but no cigar. And so how do you solve that? And so again, the solution is, again, very simple. You do this sort of screen again and again and independently derive such males. You sequence them all, and then you ask, is there a certain pattern among the genes that carry mutations across these independently derived viable hybrid males? And if there is, then
let me interrupt and just clarify. Yeah. So you mutagenize simulans.
That's correct.
You made it with melanogaster.
That's
and
correct.
then you'd get actually some males, which you wouldn't get normally if you didn't mutagenize the simulins, right?
That's correct.
But they are sterile, so you can't do any genetics.
That's correct.
So you take the one fly and you sequence its entire genome.
That's correct.
And then you repeat that over and over again.
That's correct. And in fact, this was a talent. Yep. I know.
And actually, Nitin, I'd like to interrupt too. So can you give us a sense of how many flies you had to look at before you found several that were viable?
That's exactly right. So this really talented technician, Emily Baker, who is now a PhD student at University of Wisconsin-Madison. So she and I got together and started mutagenizing these flies. Essentially what you do is you collect simulins males, you overnight feed them ethyl methane sulfonate, which is a mutagen mixed with sugar water, and then you just cross them to melanogaster females, and all you get are hybrid females that are also sterile, and you never really get males. And so we did this for 6 months. And this was a running joke between Harmeet and myself, which was we tried to do a back of the envelope calculation about how long it would take and how many such males would we need. And so my pat answer to how many males I thought we would need was 7, just an odd number, so there would be a tiebreaker if something went wrong. Mm-hmm. And so since then, Harmeet and I joked about calling them the 7 samurai. And that kind of became an inside joke that kept us motivated. And I also estimated that we would probably take 6 months, 7 males to know if this was going to work or not. And 6 months later, we had nothing.
All females, right?
All females, no real rescue males.
Mm-hmm.
In fact, that was one of the first moments of doubt. Is this even working? And so Emily and I went back and checked. Everything seemed to be working all right, but still no rescue males. Until one day, you see this lucky little male in a vial. And if you're a Drosophila geneticist, you can just look at a vial and tell if among a sea of females, you see a male. And after the first one, we got 2 more real quick. So we got our first 3 rescue males. And then we immediately rushed to the sequencer. And again, this was terrific teamwork with Jay Shendure at University of Washington and Jacob Kitzman, who is now at University of Michigan. And after the first 3 males were sequenced, we got no gene that was commonly knocked out across any three. So again, that was absolutely scary because now we had invested a year, gotten 3 rescue males, sequenced them, and asked if there are no commonly mutated genes across 3, then how in the world do we expect to get a gene that is commonly mutated across 7? But we powered on. And after the next 3 males were isolated, we now had a total of 6. We sequenced the next 3 males. And this time, we started also paying attention to deletions because that's something that we had not done with our first 3 males. And when we took that into account, the answer immediately popped out. There was a single gene that was knocked out across all 6 independently derived hybrid rescue males.
And how many flies total would you estimate that you screened through to get those 6?
So we ended up mutagenizing something like 55,000 simulins fathers. And we estimate that for more than 300,000 female F1 females that were sterile, we got about 6. So this was literally a room full of crosses, bottles and vials of unending crosses day in, day out. It was a really heroic effort, especially with Emily Baker's dedication.
So the mutagen you used, ethyl methane sulfonate, isn't that something that typically makes point mutations?
That is exactly right, which is why we weren't looking for deletions in the beginning.
You think those are randomly occurring deletions then that had nothing to do with the EMS?
No, so EMS does make deletions, but at a smaller frequency. And what had happened is that our males 2 and 3, both of them had a giant deletion in the simulins part of the genome, independent, overlapping deletions, so partially overlapping deletions on the 3rd chromosome. So that was our first hint. So once we zoomed into that region, we could see that the other flies also had point mutations or frame shift mutations or stop codons, premature stop codons in the single gene that was covered by these 2 deletions.
And what was that gene?
So this is a gene called GFZF, very awkwardly named. It mostly stands for the fact that it has a GST domain. So it's GST containing fly-wedge zinc finger domain protein. But it actually has a really fun other name called suppressor of killer of prune, which itself has a terrific history.
Tell us.
Oh, so the reason this gene is called suppressor of killer of prune again goes back to that fly room in your vicinity, which is this person Sturtevant, again, as I mentioned earlier, a giant in the field of genetics, did some crosses within melanogasters. So he found that there is a mutant strain called prune, which makes the eye color different in flies. And he found out that if you made crosses between this strain and a bunch of some wild-caught strains that look perfectly normal, the hybrids, so again, this is within species, F1s, would be dead. So he called those strains killer of prune, that when crossed to prune, gave inviable offspring. And so this is the prune, killer of prune system of Sturtevant.
Cool.
And this gene is actually necessary for that lethality within species as well.
So now you have in all 6 of your flies, which are gone because you crushed them,
Correct.
the deletion in this GFZF gene. Now I suppose you still have to prove that it is the 3rd gene, right?
That is absolutely right. So at this stage, it is a really strong candidate gene, but it's still a candidate. We really need real proof. But yeah, you were right about how valuable those 6 males were. In fact, Hermit joked that these 6 males together were more precious than either of our cars put together.
All the money you spent making them, yeah.
That's right.
And you probably have a cheap car.
That's correct. So the Fred Hutch tweeted their press release with the headline, "Why evolutionary biologist Hermit Malik and Nitin Fudness joked that 6 flies were more valuable than their cars."
Very nice.
Or something like that.
Yeah, that's great. I love that.
But the truth is, those cars are worth so little that six flies, maybe, what's the real value?
So I had to jokingly respond to that, saying that's only because you haven't seen our cars yet.
That's right.
So once we have this candidate gene, the idea again was fairly straightforward, which is, can we now produce RNA interference constructs, essentially a method to dial down the expression level of the simulins allele of this gene in hybrids, and see if that mimicked the whole rescue. So essentially, you make the same F1 hybrid fly again, which would be dead, but instead turn down the amount of transcript that the simulins gene contributes. Mm-hmm. And so again, this was fairly straightforward to design these constructs because the two alleles between melanogaster and simulins are divergent enough that you can find unique spots in that gene to target that would only target simulins and leave melanogaster alone. And if one were to do RNAi in these hybrids and ask, does this now rescue those hybrid males, then that would be great. Except another hitch there was as I mentioned to you, I was working on a bunch of things in Hermite's lab. And one of the things I was working on is the rapid divergence of the RNA interference machinery between melanogaster and simulins. So it wasn't clear to me whether hybrids would even have a functional pathway to deploy RNA interference. But we powered on anyway to see if we could see the answer. And as you know, it all worked out. RNAi does work in these hybrids. If you were to knock down the expression of simulins GFZF, that is sufficient to robustly rescue these hybrid males that would be otherwise dead, showing conclusively that this is in fact the gene.
How do you do RNAi in flies? Where do you put the RNAi?
So this is interesting. So you have a lot of tools in melanogaster that now you can borrow for these sorts of evolutionary questions. Mm-hmm. So it's actually very simple. You have a short hairpin RNA construct that you can make and drive through sequences known as upstream activating sequences, or UAS, which is actually a system stolen from yeast genetics. So you make this construct, you make a transgenic fly that now has this UAS promoter that can drive, in theory, when induced, this hairpin construct that does RNAi. And if you were to cross this transgenic strain to another strain known as GAL4 strains, when you put these two components together, GAL4 can now drive the expression of that construct from that UAS promoter. Mm-hmm. And so that's exactly what we did. We used melanogaster strains to use their GAL4 strains, make knockdown constructs in melanogaster, put those two together in melanogaster. And this is important because GFZF is actually an essential gene. Mm-hmm. If you knocked it down in melanogaster, if you put GAL4 and UAS RNAi together in melanogaster against the gene, that fly would be dead. And so there would again be nothing to cross with. Except, as I mentioned, this construct only targets the simulins version of this gene. Mm-hmm. So that's why these flies are alive. And then once we put these two components in melanogaster, we cross them to simulins males. And that was one of the happiest days of my postdocs, to see these viable flies, hybrid males, come out that we could confirm. And I can still remember the joy and excitement of seeing that result of hybrid males that are alive when generations of geneticists have only seen dead hybrid males.
But are these males sterile?
That's correct. They are sterile.
Okay, but they're alive, which is the key here, yeah?
That's correct. So this is a viability phenotype.
In the previous experiments, you don't actually see dead males, right? They don't make it through development, is that? Or do they and then die after being born? Or?
Actually, that is also a really interesting detail and wrinkle that it becomes important a little later in the story. So these hybrid males actually hatch out as larvae from those eggs. So the eggs develop, the male larva hatches out, it wanders around, eats. It's a little slow to develop. It has some defects, but it grows nevertheless. Mm-hmm. And some of them can even pupate in their race towards becoming adult flies. But somewhere during this larval to pupal transition is when they all die off.
How many days does it take to pupate?
So normally it takes about a week for a fly to do this. These guys are a little slower. But the reason this is fun, and this is again something that I didn't know despite being a Drosophila geneticist for a while, was that the way a larva develops is actually quite cool. So when the larva hatches out of an egg, it mostly eats and grows not by cell division, but by increase in cell size. So it takes whatever cells it has and just grows in size. And the DNA replicates as well, except it replicates into these what are famously known as polytene chromosomes, for example, in the salivary glands of flies, where the chromosomes just duplicate until there are thousands of copies of the same genome within the cell without cell division. And so these are polyploid cells. And the stuff that makes the adult fly is actually saved in these pockets of diploid cells known as imaginal discs. So these imaginal discs, each one of them corresponds to the adult eye, adult legs, adult wings, and so on. These are the only diploid cells, in addition to the nervous system, that stay diploid, are sensitive to cell cycle checkpoints, are regulated tightly. For example, if you were to X-ray these fly larvae, the polyploid cells just power on, they're not affected. But these diploid cells are sensitive and they can really degenerate and cause lethality at that exact stage that I mentioned, when those diploid cells need to proliferate, give rise to the adult body form. And when that cannot happen, that's when you see this lethality kick in.
Yeah, and I'm curious.
So this beautiful experiment, which you said is the most exciting of your postdoc, that was done by knocking down the simulins GFZF, right?
That is correct.
So if you do that with the Drosophila GFZF, what do you see?
That's correct. So if you did this with melanogaster,
melanogaster. They're both Drosophila.
They're both Drosophila. That is not unusual to put it that way, by the way. So it's hard to do the analogous experiment exactly the way we did because, as I mentioned, if you knock down the melanogaster copy, that melanogaster fly would be dead.
It's dead, okay.
But here we can use the beautiful toolkit available in melanogaster, and we can in fact use a genetic mutant, a knockout of this gene, instead of knockdown, do the same cross, and it turns out that the melanogaster copy, getting rid of it is not sufficient to rescue these hybrid males. It's only the simulins allele that when you get rid of, the hybrid male is alive. So there is something different between those two copies, and that's something that we can talk about as well.
Yeah, so that sent you on a search for the sequences of this gene from different strains, right?
That's correct. So we did survey a bunch of melanogaster strains, a bunch of simulin strains, and even other species to start looking at what's different. Is there any signature of adaptive evolution, something that you mentioned earlier? And the answer is yes, we do see population genetic signatures of adaptive evolution here.
How do you think that GFZF is mediating incompatibility?
So this is relevant in the context of what we know. So HMR and LHR are two genes that we now recently know, actually quite a few years after their initial genetic identification. We now know that these genes are responsible for proper centromere or pericentric heterochromatin formation. So if you knock them, so let me step back for a second. So LHR and HMR are both non-viability essential genes. So if you just take melanogaster, and if you knock out, get rid of one of these genes, HMR or LHR, the hybrid fly is still alive. And that is different from GFZF. If you knock it out, you're dead, which means GFZF is essential. These two are not. But recently, we found out that these proteins, HMR and LHR, localize to the area around centromeres or in pericentric heterochromatin. And if you knock it down, you can get some mitotic defects during chromosome segregation. But the other cool thing is, if you knock these down, you also get a lot of upregulation of transposable elements and repetitive sequences, which indicates that these genes may be involved in some sort of suppression of these selfish elements in the form of transposable elements or pericentric repeats and so on.
So can you now take these 3 genes and understand how this particular speciation occurred?
So this is what we're trying to do now. So one of the most obvious experiments that we're now in the middle of is to now put these 3 incompatible components back into melanogaster and try to recreate the whole system within the species transgenically. And if these 3 genes are sufficient to kill the hybrids, then we can be confident that we've discovered, at least on the simulins part of the genome, all the components that are necessary. If not, that would also mean that there may be additional factors, or maybe you even need a hybrid background with a hybrid suite of transposable elements and a hybrid suite of satellite repeats and so on. So a sensitized background for this to work. And this is, again, something we're in the middle of and really excited to find out.
Yeah, that's really cool, Nitin. So, I mean, one of the impressions you're left with is that you might sort of have a handle on these 3 components, and that could sort of explain all of the parts of viability here. But one of the points that you brought up and is in the paper, and Vincent mentioned as well, is that so you've sort of solved the inviability problem, but your males are still sterile. And so I'm curious if you think then that are there another 3 components that explain sterility, or could it be 1,000 components and you're sort of back in the sort of mysterious days back in the fly room where it's hard to know all of the incompatibilities on these different levels?
That's correct. That's a great question. And I think the answer is somewhat in the zone of the latter option that you proposed, which is thousands of stuff. And let me explain why we think that. For whatever reason, and we can talk about this separately as well, hybrid sterility, especially hybrid male sterility, evolves incredibly fast. So you can have 1 incompatibility, 2 incompatibility, dozens of independent incompatibilities that render your hybrid sterile evolve pretty quickly. Hybrid inviability, on the other hand, evolves slowly. So you can think of 2 isolated populations. First, you get hybrid sterility, and you get more hybrid sterile incompatibilities evolve one on top of each other. Then you get hybrid female sterility kick in, and then afterwards you get hybrid inviability kick in. So in this case, when we look at hybrid inviability, we are way past anything to do with hybrid male sterility. So I think, and I could be wrong here, but I think you would have many, many independent incompatibilities. Lots of genes make these hybrids sterile.
And so that's what's driven you in some of your other projects to the species like Pseudoobscura, where it's a much more recent divergence. And so you might be able to gain a handle on the fewer number of components that might be different.
That's exactly right. So in other words, if you were interested in studying hybrid male sterility, this would be an awful system to work with, almost intractable. That's why you go to these younger species where there's only recently evolved hybrid male sterility. And in fact, this Pseudoobscura system, we know, our work now shows that there's only a single incompatibility, just like in melanogaster and simulins for inviability. In Pseudoobscura, there's a single incompatibility, only a handful of genes involved. And that's, again, something else that's really exciting in my lab.
That's great. And then so for this specific gene here, GFZF, you mentioned it's kind of this complex, science word sandwich of different domains coming together. One of them being the zinc finger domain. And actually, Vincent and I were discussing in the last episode, TWiEVO number 3, some interesting work from Molly Przeworski's lab, where they were looking at these recombination hotspots in rodents, mouse, and also in birds involving this gene PRDM9. I'm sure you remember this, Vincent.
Yeah, sure. Absolutely.
And in that case, so the interesting connection, or maybe there is not a connection here, maybe it's just a coincidence, is that PRDM9, which also contains zinc fingers like GFZF, was found originally to be a speciation gene in mouse. Do you think there's some theme here, or is this just a strange connection or a coincidence?
This is really cool. So this PRDM9 gene that you mentioned, the wonderful work done by Molly Przeworski, It's also a gene that causes sterility between mouse species. This is decades-long work done by this guy, scientist Jerry Forage. And as you point out, the one thing that's common across some of these guys, some of these genes, is that there seems to be a DNA binding domain. So I think that is the extent to which we see some commonalities, which is PRDM9 binds DNA, overdrive has a MADF domain, HMR, LHR, GFZF. We have a handful of these speciation or hybrid sterility or inviability genes known. Somehow they're enriched for DNA binding domain. What's that about? I don't know.
So do you think that the evolution of this incompatibility, do you think it's simply a matter of you have isolated organisms and these happen to be genes that are rapidly evolving anyway, and they have to, and therefore in isolated organisms they go in different directions, so when they come back together they don't work anymore?
That's correct.
Is that it?
That's correct. And one of the sort of implicit points in what you just said is that there must be something else that drives this adaptation of these genes or this rapid ticking of these genes in their own separate populations to make them evolve quickly.
But you don't think that there's a protective reason for incompatibility, right? That it doesn't help the species to not be able to reproduce with another one. That's
correct. So this hybrid, so this is again something that Darwin himself struggled with a lot. How, if natural selection is so strong and powerful and have such a keen eye for minor variations, how in the world do you even evolve something really bad like being dead or being sterile?
Yeah.
And so the solution to that is that is actually not something that you see in nature. These independent populations and these genes adapt for different reasons. And this hybrid inviability is only an accidental consequence when these populations come back together.
Right.
So there's no selection for inviability or sterility per se. It's just adaptation going on separately, and then parts become incompatible.
Right. Okay. That's beautiful. What a great story, right, Nels?
Yeah, really fun, isn't it?
It's great.
I love it.
The thing is, I'm loving this because I'm learning so much.
You and me both.
You know,
I'm happy to be here.
This is not what I do on a daily basis, right? And so this is cool, Nels and Nitin. I started with a podcast on viruses, right, which I know and I could talk about in my sleep. And then I moved to parasites, which I don't know anything about, but I got a colleague who's a parasite. I did bacteria, which I don't know much about except using them to grow plasmids. I got colleagues. And now I've really moved out of my comfort zone, but I figured I've learned how to ask questions over the years. So,
this is going lovely. I'm loving the way you're going at it.
And I just learned so much. This is beautiful. I can't wait to keep doing this, Nels, for a long time.
Me too. And I just have to say, again, some of the themes that we were hoping to highlight on TWiEVO just in really good display here with Nitin.
This is perfect.
Right? This question that goes back to Darwin, then gets sort of chiseled at by these Drosophila guys in the fly room at Columbia. And then finally, the advent of these genomic technologies to kind of blow these questions open in a new way. Again, this idea of a functional synthesis or sort of the next generation of evolution and experimental biology coming together. Really fun.
But if the guys in 1910 had this sequencing, they wouldn't know what to do with it.
Yeah, that's right. Before we knew the gene basis or the structure of DNA and all of these things.
So, science has a sequence of how things happen. That's why it works.
Yeah.
But it's actually quite intimidating to think of if somebody like Herman Muller were around today and knew what we know today and had all these tools at his disposal that we have today. What would these geniuses work on today? And this is something that really haunts me all the time.
Yeah, it's a good question.
But the other kind of cool thing here that I also like is that we may be asking the same questions from Darwin's time or from Thomas and Morgan's time. But the gene that we found out turns out to be important for cell cycle regulation, which again brings together this contemporary synthesis that Nels mentioned about these deep evolutionary questions in genetics and genomics, and going towards understanding how the cell fundamentally works and how is it regulated.
So, Nitin, if you looked at this problem, incompatibility, in different in horses and donkeys and whatever other species are out there. Do you think there would be a theme? Do you think cell cycle control would be that theme?
I think so. So we have really struggled as a field to come up with common themes for, is there a particular developmental pathway that's labile that recurrently causes these sort of hybrid problems? And this gene is actually, as I mentioned, some of the first genes involved in two different dominant incompatibilities, one in the prune killer of prune system, and also between melanogaster and simulins. And until now, although we have just a few genes, there really hasn't been this kind of same gene showing up twice business happen yet. And this is also what makes me think that cell cycle regulation, which, again, as cell biologists think of as something that should be very, very conserved, again, throws another surprise our way.
Are there other incompatibility systems that are worked on that can reach this point where you have with flies?
That's correct. I mean, one of the things that's interesting in my field is that there actually aren't that many people studying hybrid sterility or hybrid inviability in animals that much. There are some really excellent people studying things in mouse and so on, but really it's somehow turned into a very small field. And I hope we can both make use of the idea that there's very little competition, but also acknowledge that there's wide open space and tools now to sort of go at these problems from many different directions. And the promise of always discovering new and interesting and beautiful biology stays.
I guess you have to have experimentally tractable systems. So flies are great. Mice are okay. They're a little harder than flies, right?
Right.
And then and there are probably others. Maybe zebrafish, you could do it as well, right?
Yeah, so one of the ironies of genetic model systems, and this is again something that I grappled with as a young evolutionary geneticist, is that we somehow have a knack for picking model systems that are terrible species for evolutionary genetics. So we just learned how hard it is to use melanogaster to do speciation studies. Similarly, zebrafish does not make viable fertile hybrids with any of its sister species. Same with Saccharomyces. It turns out to be now tractable, but not really convenient. Same thing with C. elegans. Again, you don't really have a closely related species that you could cross to. But one of the cool things is with the advent of new technologies such as CRISPR-Cas9, and genome sequencing and mutagenesis and all these things can be now applied to a variety of systems. Maybe not elephants yet, but there's a lot of playing space now.
Well, and it seems like this could solve one of the problems, Nitin, that you raised at the beginning of the podcast, which is that it's really hard to capture these speciation events sort of in close time to when they, or proximity of time to when they happened. And so as we're able to do more and more genomics, and then follow up with some of the gene editing technology, perhaps this opens up sort of a whole nother level of different model or non-model systems where you're just increasing the likelihood that you'll pull out a pair of species that have not been apart for too long, and again be able to tackle this question in new ways.
Absolutely. It's really a really exciting time to be here now.
Are you focusing on Drosophila in your lab, or do you move to other organisms as well?
So my lab mostly does Drosophila work, but really my trajectory has been really long-winded. So I was trained as a microbiologist, and my first love was microbial evolution, bacterial evolution. In fact, my first PhD experiment was an experimental evolution in bacteriophages. So from there, working with some yeast data sets and so on, I actually came to flies pretty late in the game. But I often tell my students to not be married to any particular species or system, but rather follow your nose to interesting questions. And so I hope that over the next 5 years, we will be doing something dramatically different.
Nice.
Nitin might even show up on TWiEVO someday.
I hope so.
Sure. Now that we know that your sound is good and you have a good connection, possibilities are endless. I think that is a characteristic of science these days, is that you can ask a question. Don't worry about the system. As long as it's amenable experimentally, use it.
Yeah,
that's correct.
Anything else from you, Nels?
I think that covers it. This was a fun paper to consider.
All right. I just wanted to end up with a couple of other things. Nels, a few episodes ago, I don't know if it was the last one or the one before, your pick of the week was the tardigrade genome sequence.
Yeah, that's right. And so here we go, an example of we've been talking for the last few minutes about how this great new technology can lead you in great directions and into interesting, weird systems like the tardigrades. And then all of a sudden we're mired in controversy. So I think I almost sort of presaged that when we talked about it last episode as my pick of the week. So these beautiful critters, the tardigrades, and this sort of exciting idea that as their genome was sequenced, I should say, that there is this just sort of massive signature of horizontal gene transfer. And that's this idea that the tardigrade genome had been acquiring genes from bacteria to the tune of something like almost 20% of the genes being from bacteria. Now, almost immediately after that paper came out in PNAS, another group that did the very similar analysis with tardigrade genomes posted on the bioRxiv, which is this preprint server, sort of a contradictory opinion that there still is a slight amount of horizontal transfer, but much more close to what you would expect for eukaryotes in general, nowhere near the 20%. And so now we're kind of in this really interesting back and forth between the two groups. It's very open. Some of the conversations are happening on Twitter as scientists sort of weigh in for or against the rampant horizontal transfer hypothesis. And it's a really fun example, I think, of seeing how science just unfolds, where people do really careful, honest work to try to put forward a cool idea, and then others do careful, honest follow-up, and then the conversation plays out. And so I would say that this is still an open question. How much foreign gene material got into the tardigrades, and we'll be following this one for the next few months going forward.
Yeah, you bet. Well, Nels, we have our first TWiEVO emails.
Yeah, pretty exciting.
And I think, since you're the evolutionary biologist, you should take that first one from Mitchell, which was actually... So that came into TWiEVO. And then we have a second one, which actually... I think it went to TWiV because I mentioned on TWiV that we were doing TWiEVO.
Oh, great.
And that writer is a TWiV person. So why don't you take Mitchell's?
Sounds good. So Mitchell writes, Dear Doctors Elde and Racaniello, I've been listening to TWiV since I started my undergraduate career 3 years ago, and I enjoy learning about all things science. I wanted to comment to let you both know how excited I am for This Week in Evolution. My high school didn't teach evolution, and any questions I had regarding evolution were answered through Darwin's On the Origin of Species, Jonathan Weiner's The Beak of the Finch, and as Nels mentioned in the first episode, Neil Shubin's Your Inner Fish. After mostly understanding the quantitative theory of evolution in 11th grade, I decided to pursue a career in science because I thoroughly enjoyed how all life is diverse yet similar in many ways. Sorry for the rambling, but I am extremely excited to listen to TWiEVO whenever it comes out, and I can't wait for future episodes. Keep on keeping on. Thanks a lot, Mitchell. Wow, that is a great message and really inspiring. I think, we hinted at this in maybe our first episode that evolution can sometimes attract some controversy when we are talking about origins of life and some of the religious and political sort of tension that can arise from that. But that's really, I think, what we're trying to do here in a large sense is to provide an outlet for bringing forth these ideas that are kind of contemporary and happening in evolution. And so I'm really thrilled to have you as a listener, Mitchell, and I hope that we can reach others in similar ways.
I have one from Anthony who writes, it's great that you will be producing an evolution podcast. Perhaps Darwin's raising of pigeons might be a topic. He sends a YouTube link, a video on YouTube link on this topic. Darwin originally had no insight into the significance of the finches in the Galapagos. It was the observation of atavistic reversion and the use of artificial selection in domestic pigeons that provided the catalyst that transformed the data collected during the voyage of the Beagle from natural history to biology. Thank you. So there's our pigeon reference, Nels.
That's right. And so stay tuned, because I guarantee we'll be talking more about pigeons, which, again, sort of echoing or mirroring Nitin's story of taking these old questions and then re-examining them in contemporary ways. There's another fellow here at Utah by the name of Mike Shapiro who's been doing this for the last decade or so. He grew up as a stickleback evolutionary or developmental evolutionary geneticist, and has moved a lot of his laboratory and most of his attention to pigeons that have been bred in very sophisticated ways for morphological traits that sort of, were interesting to pigeon breeders. And now Mike is using some of these genomic techniques and tools to get at the genetic basis of this, and he's finding some really exciting and interesting things. And so stay tuned, Anthony, and thank you for bringing up the pigeons.
Would he be able to, your colleague, would he be able to provide some insight into the Darwin pigeon story?
Yeah, in fact, so if you catch Mike Shapiro on the road giving a seminar or in lecture, he has really gone back to some of this history and pulled out some of the original texts and some of the illustrations of the pigeons that Darwin was sort of ruminating over a long time ago. And then he's now, combining that with his work in cool ways.
Cool.
Yeah.
I think we got more Darwin coming up here. Let's do some picks, Nels.
We do. We're on the really hammering home the old classic evolutionary chestnuts here. So my pick of the week is a watercolor of Darwin and some of his crew on the deck of the HMS Beagle on one of his voyages. I believe that this watercolor, this piece of artwork, was just sort of hiding in a private collection and was almost just rediscovered very recently. And then it was actually just auctioned in the last few days by Sotheby's and went for about $75,000. So I've got a link up to that watercolor, and it's really cool because you can zoom in and you can see sort of this caricature, cartoonish scene on the Beagle and some of the collections they were doing, and there's even captions, sort of a conversation playing out in this watercolor.
I heard you were the buyer of that.
Yeah, my assistant professor salary is not quite that lucrative, unfortunately.
You should try trading my car for it.
Yeah, there we go. I think I'll need 12 of Nitin's flies before I can make a purchase of that level. How about you, Vincent? I think you have a very interesting pick.
So I have a pair of articles on SpaceX. So one of them's in The Verge, and the other's in Ars Technica. So SpaceX is a company founded by Elon Musk, who's the guy who owns the Tesla company. And they've been, they want to commercialize rocket launching. In fact, there's another company started by Jeff Bezos, the owner of Amazon, or I don't know what you call him. He doesn't own it, but he's I don't know what
he's got definitely a vested interest, right?
Yeah, so he started a company called Blue Origin. And they both have the same goals. They want to make rocket launching commercially viable, give people trips, bring stuff up there. And the key here, one of the keys, is to be able to reuse the rockets, not just put them up there and have them burn up. So they want to land them. And if you remember all those old science fiction movies of years ago, the rockets would land, they'd be coming down with the flames roaring out and slowly coming down, gently touching the ground. So these guys, SpaceX, finally did it. They've had a few failures.
Cool.
Monday night, they launched a rocket, and then it settled back down on a landing site, and it didn't blow up. It didn't fall over. And on the which article? The Verge. They have a movie of everybody in this control room going crazy, when this thing happened, which is pretty cool.
Yeah, really neat.
So this is going to be the future of rocket launching. The NASA's finished. They had a big contribution to make, and now these private companies are going to be doing it. They'll figure out how to get it to work reliably, and it's going to be cool.
Yeah, that sounds like one of the sort of transformative events for that field was that SpaceX was able to pull that off. I think that gives them some real credibility moving forward.
Reminds me of all the attempts we made to launch a rocket, and there are videos you can find of rocket after rocket either taking off and blowing up or rising a few feet and then coming back down and blowing up. Right? You remember those?
Oh, yeah, definitely. And it's sort of a good metaphor for the biological laboratory starting experiments, right?
That's right. You crash and burn a bit.
Exactly.
The funny thing is that up in the first human we sent into space was Alan Shepard. And up until not too long before his launch, these things were still crashing.
Yeah.
And it's amazing that his worked. And so many after his worked as well. They really got it,
The bravery. Yep.
That's correct. It's kind of interesting that the Blue Origin, Jeff Bezos' rocket that they launched and brought back for the first time is called the New Shepard.
Yes. You think it's named after Alan Shepard?
I don't know. I wonder that. But that's a cool reference.
Yeah, so Blue also did a similar thing, right? They launched and landed, right?
Correct. But this was, for them, it was just a simple go up and come down without actually any horizontal velocity or anything like that. So.
Right. Yeah, so it's pretty cool. Nitin, I hear you have a pick.
I was actually really excited about this Falcon landing, but I found another pick for this week in evolution. But you didn't mention the year; you just said week. So I'm going to go back to 1895 to this day, December 22nd, where Wilhelm Conrad Röntgen took the first X-ray photograph of his wife Bertha's hand. It's a really famous picture taken today 120 years ago.
Wow. I'm surprised this is not the Google Doodle for today.
Yeah, really.
Yeah, it should be. But no, that started the whole radiology or radiography field. So
yeah, I just found that famous picture. She has a ring on, I think.
That's correct.
Nice. Oh, that's great. Yeah, we don't care when the year is, as long as it's just that we like it. And of course, we launched TWiEVO. Was it last week, Nels? I don't remember. 2 weeks ago.
That's right. The first episode is live and kicking.
So, Nels, I'm thinking of releasing them, the ones we have, every week or two. What do you think of that?
I think that's a great idea.
Because people will like that. And so episode 2 I'll release this weekend.
Fantastic.
People are hungry for more.
Well, this is, it's not viruses, bacteria, parasites. This is a field in a way, right? It's a question, as you put it, Nitin, not just an organism.
That's correct.
And I think it's got a lot of appeal because of that, and we're going to cross all kinds of organisms. And if you don't like viruses, you'll find something on TWiEVO that you like.
That's right. Just come back to the next week in evolution.
That's
Although
right.
I think will spend a fair amount of time in microbial territory, since these are both sort of historically, but also today, the systems where you can really dig in and do a lot of experiments. And so one of the sort of powerhouses are broadly defined microbial populations and considering their evolution.
And you can find TWiEVO at iTunes. Apple was amazingly fast at approving my submission. I submitted it on a Saturday, I think. Yeah, and I think they approved it on Monday.
Oh, fantastic.
And I'd like to think that our track record meant something, but probably not. They're just probably efficient. So it's on iTunes, and you can find the iTunes link at microbe.tv/TWiEVO. And of course, if you're on a mobile device, either iOS or Android, you can use a podcatching app to download all of the episodes, subscribe and download. And we do want to get your questions and comments. We also want to get your picks of the week. We'll do listener picks. You can send them to TWiEVO, TWiEVO, This Week in Evolution at microbe.tv. I like the name TWiEVO now. It really has a good ring.
I agree. I'm starting to fall off the tongue pretty regularly. Yeah, pretty good.
Nels Elde is at cellvolution.org. You can also find him on Twitter. He's L Earlybird. Thank you, Nels.
Thank you, Vincent. This was fun as usual.
I also should say that you're at the University of Utah, of course, but maybe your social media links are more important.
Equal.
Well, the university pays the bills, right?
Exactly. That matters.
And our special guest today, Nitin Fudness. He is also at the University of Utah, and he's got a website called flygenetics.com. Thank you, Nitin.
Thank you so much, Vincent, and thank you so much, Nels. It's lovely to be here in your esteemed company, and it's a privilege to be on TWiEVO.
Oh, thank you. You were really great and engaging, and I hope our listeners love it. I certainly did, and we'll have you back again when you find another gene.
Thank you. I'm excited about it, but don't hold your breath.
Things go slowly.
Yes, but there'll be other things to talk about, I'm sure.
Absolutely.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.